Thanks to you all for coming. I'd like to present my dissertation research, which is titled Ecological and Evolutionary Drivers of Morphology and Alginate Content in Giant Kelp, and I'll be focusing on that alginate chapter for this symposium. So in case you haven't seen one before, this is a giant kelp forest underwater. And this one is from a kelp forest I visited in Chile. And as you may know, kelp is a really important habitat forming species, and it creates kelp forests, which provide a home for lots of fish and invertebrates and feeding grounds for birds and marine mammals. But the kelp itself is also a really important natural resource. So it's harvested and cultivated around the world for extractions of chemicals for various products like pharmaceuticals, can be the soft coating for tablets like probiotics, can be food for humans, not just people, not just animals. Supplements like iodine is popular. Some kelp is farmed for production of biofuel, and then some chemicals can also be used in biomedical materials like wound dressings or dental molds, and others can be gelling or thickening agent in cosmetics or food. And then the kelp itself can also be a fertilizer since it contains a lot of important nutrients. And one of the biggest global producers of harvested kelp is Chile. And during my first year of graduate school, I got the opportunity to go to Chile as a Fulbright scholar. And it was through my experiences in Chile working with kelp that I got the inspiration for my PhD research. So I was stationed in a small fishing town called Las Cruces, and I stayed at this really gorgeous marine biology research station, which looks a lot like the coast of Santa Cruz. And for my project, I was traveling around the Central Coast, interviewing fishermen who harvest kelp and learning about their different methods and sort of how they perceive the future of the fishery, the kelp fishery. And as I started to learn more about kelp and look at it up close, I noticed that this giant kelp in Chile that I was seeing looked a bit different than the giant kelp I'd seen in California, even though they were supposed to be the same species. So in Chile, lots of times I would walk along the beach and see that there's the kelp washed up on the shore. And a lot of times you would see this basal structure of the kelp, which is called the holdfast. And so I'd kind of pick that up and look at it. And what I was seeing in Chile were kind of these long, flattened, elongated holdfasts, whereas in California, I'd seen more of these large mounding cone-shaped holdfasts. And so it turns out that these are actually two different ecomorphs of the same species, giant kelp, and that globally, there are actually 4 ecomorphs of giant kelp. So by far, the 2 most common are Peripera, whose distribution is colored in blue, and Integrifolia, whose distribution is in orange. And then the other 2, Angustifolia and Laevis, have much smaller ranges in the Southern Ocean. And so these ecomorphs were once thought to be actually different species, but they were then lumped together as the same species since researchers found that, at least in the laboratory, they were able to interbreed, and previous genetic work wasn't able to find any differences between these ecomorphs. So these ecomorphs were known, but the actual mechanisms driving the variation in morphology in that holdfast form was still unclear. And so the main differences between these 2 most common ecomorphs, the Peripera and Integrifolia, the main difference is in the holdfast. So from the surface, they actually look pretty much the same because all you can see from the surface are the blades, which are those big leaf-like structures, and the stipes, which are the long kind of stems that all the blades are attached to. But down at the holdfast, the Peripera morph has this cone-shaped, mounded holdfast, and the Integrifolia morph has an elongated, flattened, strap-like holdfast. And also in the Peripera morph, all of the stipes are kind of emanating from one central location. And in Integrifolia, the stipes come up at different spots along the holdfast. So those are the main morphological differences. And here's what they look like in real life. And so for my dissertation, I wanted to understand what are the driving forces behind these morphological differences? And are there any functional consequences in the kelp from these different morphologies? And so kind of the main theory prior to my research about how these different forms came about was that the holdfast morphology was determined by the type of environment the kelp happened to be growing in, specifically the depth. So the theory was that if the kelp happens to be growing in deeper water, it'll have a porifera shape. And if it happens to be growing in shallow water, it'll have an integrifolia shape. And if it's growing maybe at intermediate depth, it'll have a hybrid intermediate shape. And so this is why they were called ecomorphs, because the definition of an ecomorph is a local variety of a species whose appearance is determined by its environment. But this had never actually been tested before. And so my research actually found that this is not the case, that the holdfast morphology is actually genetically determined and not changeable by the type of environment that it's in. So that's kind of just a little teaser into my dissertation, which consists of 3 chapters. In the first chapter, to answer these big questions, why these morphologies came about, I did a population genetics study where I looked to see if there were any patterns of the giant kelp ecomorph distribution and then their genetic differentiation. So I sampled 18 different populations across a large chunk of their distribution in Chile, and then also some sites in California. And then I did used whole genome sequencing to compare these individuals. And then in my second dissertation chapter, I wanted to experimentally test the influence of environment versus genetics on morphology. So I did an experiment where I grew both of the morphs in the lab from their microscopic spore stage. That's what you're seeing in the little petri dishes of strings with little bits of kelp. And then I outplanted those different morphs to concrete parking bumpers that we put at the bottom of the ocean. That's what you're seeing in this next picture. And then on the bottom picture, you can see the kelp grew really large, and I was able to make some morphological assessments. And then in the third chapter, which is what I'll be focusing on today, I wanted to see what are the functional consequences of these different environmental and genetic factors on one of the most commercially important chemicals that giant kelp produce, which is called alginate. So alginates are structural polysaccharides in the brown algae cell. So on the left is pictured of some dried alginate fibers from one of my samples. And alginate has lots of varied applications in different industries, from pharmaceuticals to cosmetics and foods and biomedical materials. And actually the global alginate market is projected to be worth over $1 billion in the next 5 to 6 years. And so this is a large part of the overall seaweeds product market. And since alginate is a structural component of kelp, I thought that it might also relate to morphology. So something important to know about alginate is that it's composed of these 2 types of uronic acid blocks, that's mannuronic acid and gouluronic acid, or M and G. And so these 2 blocks link together to form the alginate polysaccharide. And the proportion of M to G and the way they're linked together determines the physical properties of the alginate, and then also of the kelp tissue that the alginate came from. So if the alginate has more gouluronic acid blocks, it'll produce a more rigid gel that can be used for pharmaceuticals and foods. And if it has more mannuronic acid, it'll produce more elastic gel that's good for textiles and wound dressings. So there is utility for both of these kinds of alginates in industry. It just depends on what you're making. So my research questions in this chapter were to see, how does alginate composition, meaning the Mg ratio, differ between giant kelp ecomorphs and across different locations? And how does local environment influence alginate composition? So I hypothesized that alginate composition would differ between the 2 ecomorphs, and that alginate composition will vary across different environments, even within the same ecomorph. So to answer this question, I sampled 12 populations in Chile, 6 porifera and 6 integrifolia. And at each site, we collected blade tissue from 5 different individuals for a total of 60 samples across the 12 populations. And the sites varied in depth from subtidal to intertidal. So sometimes we collected using scuba diving, or other times we snorkeled. And then after collecting those blades, we dried them, and I was really lucky to be able to partner with this kelp processing plant just outside of Santiago in Chile, where they gave me space to lay out all my kelp to dry in their nice hot and dry climate. And then I collaborated with researchers at the Norwegian University of Science and Technology to extract alginate. So with the dried alginate or from the dried kelp, I used an acid-base extraction process to extract out the alginate and used ethanol to precipitate out the fibers. And then from these fibers, I could quantify or characterize the composition using nuclear magnetic resonance. And so this large machine outputs this kind of spectral output for each sample. So shown here is the output for a single sample. And by knowing the location of the different M and G blocks along the polysaccharide, we can integrate over these peaks and then get a proportion of M to G blocks in each alginate sample. And importantly, since the M and G are the only two components of alginate, their proportions always add to 1. So I'll just be referring to proportion G in my analyses since M is just the inverse. So first of all, let's look at some of the environmental conditions at the different sites. So the Porphyra and Integrifolia morphs are geographically separated. So they occur at different latitudes, basically no overlap. And so since sea surface temperature tends to correlate strongly with latitude, it's not surprising that we see that the ecomorphs are pretty diverged at temperature, with the Porphyra in blue being at slightly cooler temperatures than Integrifolia in orange. We can also see there's also on the y-axis a lot of variation in depth, regardless of ecomorph or site location. So in both morphs, we're seeing some individuals that are more subtidal and some that are more intertidal. So to evaluate how much the alginate composition was driven by morph alone rather than environmental factors, I created a model using morph and sea surface temperature and depth. And so here I'm showing the least squares mean proportion guluronic acid. So this is the proportion of G in the alginate after accounting for differences in G driven by the environmental variables, sea surface temperature and depth. So here looking at the influence of morph, we see that the Integrifolia morph has on average 49% G, whereas Porphyra has on average only 40% G. So this is about a 25% difference in mean guluronic content. And so when you scale this up to industry level, the morphs may have potentially very different applications depending on the type of material that's to be created from the alginate. Now let's look at some of the environmental variables. So here, this is a model plot modeling the relationship between depth and G after accounting for the effects of morph and sea surface temperature. So we see that G content is negatively related to depth, where shallower kelp towards the right end of the x-axis tend to have lower G content. And this may be adaptive, since shallow areas tend to experience harsher wave force, with waves crashing right there. So it may be better to have a low G, high M alginate ratio, meaning that the tissues would be more elastic, more flexible, and more likely to bend in the waves rather than break. We can also see that G content decreases at higher sea surface temperatures. And this is kind of interesting because if you'll recall, the Integrifolia morph tended to have higher G content, but were typically found at the higher sea surface temperatures. So it's possible that if you were to grow Integrifolia at lower latitudes with lower temperatures, maybe you could maximize G content in the whole species. So in conclusion, we can see that the Porphyra and Integrifolia morphs have different alginate compositions, which may make them suited for distinct industries. And alginate is also related to depth and sea surface temperature. So it may be important to consider both morph type and local environment when you're considering the application of the alginate that you're making, either from harvesting natural seaweed populations or cultivating in aquaculture. And in general, it's important in aquaculture for aquaculture of kelp to understand the natural history and the ecology and the chemistry of this species for successful culture. And that kind of segues nicely into my future research, where I'll be a postdoctoral scholar at Woods Hole Oceanographic Institution, which is clear on the other side of the country in Massachusetts. And I'll be working on sugar kelp aquaculture research for optimizing different traits for food, biofuel, alginates, and other things. And I would like to thank lots of people who helped with all aspects of my dissertation, including those first two chapters, which I didn't get to talk about in detail today. But I want to highlight the folks who really helped with the alginates work, which were my collaborators in Norway, Finn, Olav, and Venka. And I also really want to thank my advisor, Pete Ramundi, my committee members, Sylvan, Christy, and Dan. And also special thanks to everyone, all the staff in the EB department and the CSP staff as well. And if you have any questions, I'd be happy to take them. Thank you all so much for listening.